ফার্স্ট হাফ কি ডক্টর সন্দীপন মল্লিক দাশগুপ্ত স্যারের, সন্তোষ স্যার?
হ্যাঁ,
ওনাকে কি ফোন করতে হবে?
আব্দুল স্যার করেছেন কিছু।
আচ্ছা। सर मैं सबको एडमिट कर रहा हूँ। হ্যাঁ করে দাও, অলরেডি 10টা 50 বাজে।
Welcome to BPT, Bharat Pharmaceutical Technology, a premier institute committed to pharmaceutical education and research. At BPT, we offer different academic programs like Diploma in Pharmacy, Bachelor of Pharmacy, and Master of Pharmacy in Pharmaceutics Specialization. Equipped with a well-stocked library with over 6,500 books and 400 journals, along with a dedicated reading room. With experienced and supportive faculty members, PPT stands as a beacon of knowledge,
Yes, sir.
innovation, and service in the evolving field of pharmacy. Our industry-aligned curriculum is continuously updated to reflect the latest advancement in medicine and technology. We emphasize hands-on training, research to provide a robust foundation in pharmaceutical science. Our state-of-the-art laboratories, equipped with modern equipments, is designed in shaping competent pharmacists ready to make a difference in the global health. Our students thrive in an environment that promotes innovation, critical thinking, and professional excellence. Join us, grow with us.
Trial. 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 Trial.
Pralay sir, please mute this backdrop sound. হ্যালো, হ্যাঁ একটা সাউন্ড হইতেছে ওই স্যারের লগে। এখন দীপন স্যার একবার একটু ইয়ে করো, চেক করো সব ঠিক আছে নি? ও
Good morning. Good morning, everyone. Sir, Sandeepan, sir.
Good morning, sir.
Sandeepan, sir. Shall we start the session?
Trial.
Sir, your voice is not audible, sir, properly.
Trial.
Sir, shall we start the session, sir? Sandeepan, sir.
The sound, there is, I think, some sound related issue.
Trial.
Sir, it's actually vibrating. The sounds are not coming properly. It's not coming, sir.
Sir, there is some issue with sound, sir. Your voice is not audible.
Trial. 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 Trial.
Sir, you are not audible, sir. No, sir.
Trial.
Your mic is not on, sir.
Trial.
Yeah, good morning, Sandeepan sir. I think if you are using laptop, you can use without earphone, no issues. I think laptop audio will work.
Trial. Trial.
कौन है भैया?
Very good morning. Uh, Participants are requested to stay with us. There is some technical glitches in the sound system, and our honorable speaker sir will be here with us soon. It will be resolved very as soon as possible. Thank you.
Am I audible?
Yes, sir. Yes, sir.
Yes, sir. Yes, sir. You're audible.
Sorry for the inconvenience.
No problem, sir.
This is
No
the
issues.
problem, online things.
Bhaskar sir, please continue. Start.
Shall we start, sir?
Okay, sure.
Yes.
Namaskar, everyone. A very good morning. After the completion of these 2 successful days of our EFDP program, we have stepped into the 3rd day today. So I would like to take the privilege to welcome our esteemed guest, eminent speaker, and active participants to this EFDP program, which is entitled as Immersive Technologies and Artificial Intelligence in Pharmacy Education, Building the Future and Learning, organized by Bharat Pharmaceutical Technology, Agartala. So on behalf of the organizing committee of Bharat Pharmaceutical Technology, I, Bhaskar Jyoti Sharma, Assistant Professor, heartily welcome you all to this program. We are fortunate enough to have with us today an eminent and incredible speaker, Dr. Sandipan Das Gupta sir, HOD, Department of Pharmaceutical Technology, Macau, West Bengal. So now I would like to request Dr. Asmita Shah ma'am, Associate Professor of Bharat Pharmaceutical Technology, to kindly grace our speaker. Ma'am, over to you.
Good morning, one and all. It's my pleasure to have the opportunity to introduce our today's speaker, Dr. Sandipan Dasgupta. Dr. Sandipan Dasgupta is an accomplished pharmaceutical academician, researcher, and administrator with over 16 years of teaching and 3 years of research experience as a UGC fellow. He is currently serving as head of the department at Maulana Abul Kalam Azad University of Technology, West Bengal. Dr. Dasgupta holds a PhD in pharmaceutical sciences from Dibrugarh University, Assam in 2016, MPharm in 2008 in pharmacology. His academic journey has been enriched by industry exposure, having worked at Cipla and Unichem Labs, where he gained valuable insights into pharmaceutical manufacturing, regulatory compliance, and quality documentation. Dr. Dasgupta's research expertise spans pharmacology, nanomedicine, drug delivery system, and cancer immunotherapy. He has authored 24 peer-reviewed publications in reputed journals and contributed to over 10 book chapters with leading publishers. He has also authored a book on gut microbiota and disease pathways, reflecting his growing interest in microbiota research. In addition, he holds 2 patents, one on lipid nanoparticle-based antitubercular drug delivery and another on biomedical device for inducing emphysema in animal models. Beyond research, he has delivered invited talks at various national and international conferences. A committed teacher, researcher, and academic leader, Dr. Dasgupta combines academic excellence with industrial know-how and administrative acumen. His vision is to advance pharmaceutical education and translational research in India, with a focus on nanotechnology-driven therapeutics, cancer immunotherapy, and microbiota-based interventions that address pressing global health challenges. Thank you and welcome once again, Dr. Sandipan Dasgupta, sir.
Thank you, ma'am. Thank you for my introduction. First of all, good morning to all of you. And thank you, Dr. Debnath and other faculty members for inviting me and given me this opportunity to present this topic. So thank you. So shall I start the session?
Yes.
Yes, sir.
Yes.
Yes, sir. Yes, sir.
Yes, sir. Yes, sir.
Okay.
Sorry, your voice is not audible, sir.
Am I audible, all of you?
Yes, sir.
Okay. So good morning, all of you, and good morning to Dr. Devnath and other faculty members. Thank you for inviting me and giving me this opportunity to present this topic in the FDP. The name, that is Immersive Technologies and Artificial Intelligence in Pharmacy Education, Building the Future of Learning. So my topic is Artificial Intelligence in Pharmaceutical Technology and Revolutionizing the Cancer Immunotherapy. Nowadays, the artificial intelligence is one of the biggest thing which is come into act everywhere, not only in pharmaceutical industry, but also in various areas. Actually, this artificial intelligence is transforming the pharmaceutical technology and it playing a revolutionary role in cancer immunotherapy. AI is no longer a science fiction, actually. It is a real world tool that changing how we discover drugs, how we personalize a treatment and fight deadly diseases like cancer. So in my next slide, slide is visible to all of you?
Yes.
Yes,
Sir.
sir.
Okay. So John McCarthy is a biggest scientist from USA and is the father of artificial intelligence. He introduces this artificial intelligence and its effect and the advantages. Now, first of all, we should know what is artificial intelligence. It's simply ability of a machine to perform a specific task that usually require human intelligence. That includes learning, then reasoning, problem solving, and decision making. Now, what is the importance in pharma industry? How this AI is very much entered inside the pharma industry. This pharma industry or pharmaceutical field is normally generates lots of data, complex data, that is genetic data, then clinical trial data are there, various patient records, then chemical structures. All these things, collecting those data, storing those data, and help those data to make sense of it. Now, how it can be utilized? That is the main aim. Now, nowadays, we are using artificial intelligence. One of the common example, that is ChatGPT. We are using this artificial intelligence, like we are improving the language, we are improving a writing types, we are giving a set of data to analyze those data, and from that we are taking the final inference. That means our work is reduced. AI can reduce the all over the work, as well as it can reduce the time taken by human being. Maybe that human being, for example, 10 to 100 people are involved in collecting those data and then summarize those data. But here we can see that AI can reduce all those efforts and bring a result from that. For example, when in clinical trial, the data what we are getting in clinical trials or R&D, that will takes longer time to collect those data and to interpret those data, and sometimes there are so many errors can happen, manual error, that we can avoid. Now, why AI technology is needed in pharmaceutical technology? Now, as I told you that accelerating the drug discovery process, enhancing personalized medicine, optimizing manufacturing and quality, and traditional drug discovery challenges. Now, to develop a specific molecule, it takes about 15 to 16 years, a long time. And behind that, we are using or spending lots of money, millions and millions of money. And finally, once the drug came into the market, if we are getting any ADR or severe ADR, we are withdrawing from the market. So the whole process, the whole time, whole manpower is going into the vein. No use of that. The total duration, the 15, 16 years, gone into the wastage. So here we can use AI. AI can reduce the timelines, the duration, and as well as it can reduce the failure rates also by predicting the good things, by predicting extra inferences from those data, which a human kind or human being cannot be done. So these are the AI advantages like data-driven predictions, rapid identification of drug candidates, and real-time optimization. Now, as I told you that there are several types of advantages we have seen in AI in pharmaceutical industries, like reduced time and cost, as I told you, then high predictive accuracy, improved target success rate. Okay, so these are the improved regulatory compliances. All these things can be achieved by using AI. Normally, traditional drug discovery is very slow, very expensive, and high failure rate was seen. On average, as I told you, that 10 to 15 years, billions of dollars at wastage, and to develop a single new drug, and most candidates fail along the way. So here, this drug discovery process, this AI can be used to accelerate the drug discovery, to personalize the medicine, to improve the manufacturing and quality controls, and all these things. And in future, we can predict the market will increase up to $9 billion about in by 2030. It is expected that nowadays slowly AI is used in various phases and various aspects, and the market value will increasing go and day by day. What are the key drives of this pharmaceutical industry? As I told you that mainly it can identify disease very accurately, fasting the drug discovery process, maintaining consistency in production, ensuring quality controls. In pharmacovigilance, detecting adverse drug reaction from medical reports and even social media also. And in clinical trial, recruiting the right patients, reducing dropouts, and monitoring outcome in real time. So nowadays, we need personalized medicine. That is, what is personalized medicine? Actually, person to person, there will be a genetic variation. There are various type of person, their genetic variation is different and their location is different. So for that, their food habit is different. So depending on that, there is a changes in enzymatic production, and that leads to that needs personalized medicine. Now, what are the AI technologies or tools are used? Here you can see that machine learning, that is predictive modeling for bioactivity, which can predict the drug activity. Then deep learning process, that is solve complex biological problems. And neural language processing means knowledge from text, literature, patients. Then AI tools, various AI tools are there. IBM Watson for drug discovery process. IBM developed a AI tools, that is Watson, and this is used for drug discovery process. Then benevolent AI-driven drug discovery process, in silico medicine, de novo drug design, and then Atomwise AI for structure-based screening. Now, how this health outcome that matters to the patient. This, in previous slide, I have shown you that, 1 minute. Here you can see it represents by rising the graph. The graph is rising, focus on measuring improvement in patients, in healthcare system, and well-being rather than just the number of treatments and procedures. Then this next one represents key, that is ensure all patients, regardless of socio-economic background, location, or demographic, have access to high quality cares. This is the key, symbolizes the unlocking healthcare for everyone, fair, inclusive, and accessible. Next one is that represents by the database, involves using research, digital health, data analytics to improve decision making, and data helps track outcomes, then predict disease trends, personalized treatment, and support evidence-based medicine. Next one, that is digitally enabled care, that is represented by the computer and cloud. Digital looks like tools, that is like a telemedicine or cloud platforms or remote monitoring, makes care more patient-centric, continuous, and convenient. to all. Now, these are the challenges what we have to face when we are using AI. One is data issues, lack of standardized and high quality data sets. Still, we don't have a standardized format, a standardized data collection procedure. That is the limitation. Then interpretation of this data. Sometimes lots of data are collected in different sources, but interpretation of various kind of data has become a very complex process. Then regulatory barriers are there. There is no specific rules is available with FDA or EMA, or any guidelines is still not that much developed. So as it is not developed that much, there is a barrier in regulatory issues. Then ethical concern. There are bias in algorithms and data privacy. Biasness can be produced and it will vary one person to another person. For example, a person from India or a person from Sri Lanka or other country, their genetic is different from our country, their food habits and their other things. Enzyme expression is different, and types of diseases are also different. So leads to that there is a biasness. The data which is collected from India or the data which is collected from Sri Lanka or other country, that will be produce a biasness, because artificial intelligence only follow a specific pattern. And that's why, for example, the program is made for a specific disease, but that program is not made for a various kind of area. They are not able to identify the differences in person to person. They are giving a generalized form of information that produces a biasness among the data. These are some case studies are there. That AlphaFold DeepMind, this is used to revolutionize the protein structure prediction. Modern AI that accelerate COVID-19 mRNA vaccine during the development of COVID-19 vaccines. This AI technology is used very much. The Novartis Microsoft partnership, AI-powered digital drug discovery labs are there. They are also now comes to another thing, that is Varj Genomics, then Santen and 2XAR to identify new drug candidates, then HealX and Deep6AI. All these AIs are developed in various companies. For example, Varj Genomics. Actually, this is a biotech company. It was started, I think, 2014 or 15, and it accelerates the human multi-omics data to accelerate the drug discovery process. It can build large proprietary human disease tissue database, and use of this AI platform convergence, that is to identify the novel drug targets, and as well as it will focus mainly on neurodegenerative diseases like ALS and Parkinson, etc. Next, another company, that is Santen and 2XAR. These are the two companies. They are collaborating with each other to work on glaucoma. That Santen is a German, I think, Japanese company, or 2XAR is a US company. They are collaborating with each other and they are working on glaucoma. Next one is Helax, another biotech company from UK. They are normally focusing on rare diseases. The diseases normally the pharma companies are not concentrating on those diseases. So these rare diseases, to identify those rare diseases, their treatment, these companies are working, as well as repurposing of drug. Repurposing of drug we have seen in COVID-19 also. So this company is working on these two aspects. Then Deep 6 AI. This is actually US based company, and this is also used to identify or use the repurposing of drug or like that. Actually, these are the companies working on this things. Now, application in AI pharmaceutical technology, as I already told you, that disease identification, drug discovery and drug development process, manufacturing, pharmacovigilance, clinical trial, all aspects, these things are used. Now, next we can come to the cancer immunotherapy. There are various kind of immunotherapy available. So AI is also working on this. It is normally this cancer immunotherapy is a breakthrough treatment that use the body's immune system to fight against the cancer. Example like checkpoint inhibitors, CAR T cells, cancer vaccines, all these things. How that AI helps here? AI actually analyzing the patient genetic and tumor data, then predicting who will respond to which therapy, and optimizing treatment combinations. In short, if I want to tell that AI helps to match the right patient with the right treatment. AI integration in cancer immunotherapy workflow. Normally, we have to give the patient data. Then we have to identify the biomarker or discovery of biomarker. Later, we can outcome is predicted and what kind of model should be there, that is shown in this picture. And finally, we are getting the treatment.
1200 টাকা লাগে সারা বাড়ি মানে মোটামুটি 22
Now, next slide here, you can see that role in cancer in immunotherapy, that is target identification. That is most important thing, that AI algorithm analyze vast genomic and proteomic data to identify novel tumor antigens and immune checkpoints, leading to more precise therapeutic targets. Here you can see that there are four things are there. Epigenetics. Epigenetics is the study of heritable changes in gene expression that occurs without altering the DNA sequence. So, for example, DNA methylation, all these things. And genomics, as you all know, that analysis of complete set of genes. Proteomics is the study of proteins, okay, cell, organism, like that. And metabolomics, that is the comprehensive analysis of small molecules that reflect cellular process and biochemical activities. All these four are called as multiomics. This is the combination of epigenetics, genomics, proteomics, and metabolomics. This can be analyzed by using AI. So you can imagine that how the AI is summarize the whole process. That multiomics integrate different biological data sets. And after that, it approaches to network-based and machine learning-based. Network-based means, for example, degree of centrality to find a key gene, and machine learning based, that is neural network, that is to detect the complex pattern. And finally, it will come to the application portion. Finally, it will produce like anti-cancer targets or druggability or drug screening or drug properties. Anti-cancer targets identification, finding that specific gene through that AI where you have to target, where the drug can be targeted, and a specific treatment approaches can be prepared. Druggability assessment, that means targeting can be treated with a specific drug, whether the target can be treated with the drug or not. Then identifying the effective compound, which compound is specific or which compound is required for that specific target by using drug screening. And finally, drug property analysis, this toxicity, side effect, all these things can be analyzed. So all these processes can be analyzed by AI. So the whole process is summarized and sorted as compared to the human being. If the human being is involved in this whole process, it takes longer time. Same here, the previous one, that biomarker discovery, as I already told you, how the AI is involved in it. Now, AI role in cancer immunotherapy. Next one is drug discovery and development process. As I told you in previous slide in the beginning, how it reduces the drug discovery process, how it reduces the time consumed by the whole process. This figure actually showing the artificial intelligence supports drug discovery and cancer immunotherapy. One is data inputs. AI uses diverse kind of data. They are using different types of sources like clinical sequence, then experimental results and molecular structures. Then algorithm, this data are processed into molecular features, trained and tested with neural networks to make a specific prediction, which kind of drug it is or which kind of target it is that can be possible. Now, finally, it comes to how AI helps in disease diagnosis by identifying the target, identifying the therapeutic targets. They are doing the computational screening. After the computational screening, they are predicting that the drug can be targeted in that specific area. Here, the things are given in short in picture, that preclinical drug discovery process, then clinical trial, how it can be made shorter, then recruitment of trial patient. Here also you can use AI to select random persons or patient into the data by analyzing their age, sex, is there any other disease are present or not, or they are taking other drugs or not, concomitant administration is there or not. That all these things can be used by AI technology. And finally, clinical data analysis, whatever the data we are getting, we are utilizing those data. Here you can see that AI in personalized cancer immunotherapy. Patient data, then genetic profile and historical outcome. All these three, considering all these three, that artificial intelligence finally personalized treatment recommendation can be given from that AI. For example, they are collecting the patient data in different patient, then genetic, they are studying the genetic profile, they are taking the history of that specific patient, is there any other disease are present or not, or they are taking any other drugs or not. Everything they are studying, age, sex, body weight, everything. After that, they are collecting data and preparing a specific set of algorithm to find a specific treatment approach. Here in immunotherapy also, oncologists are reviewing those data and patient consultation, all these things. After that, that specific target can be planned, and according to that, the treatment can be given. Now, here we come to there are, as AI having some advantages as well as some disadvantages also. So some opportunities are there and there are some challenges are there. Now, here we can see that precision medicine is an approach to a healthcare where treatments are tailored to the individual patient based on their genetic makeup, lifestyle, environment, all these things. It is not like that normally what we are doing. The same drug is given to various type of people. The old age, geriatric patient, middle-aged patient, child, woman, the same medicine is given to all. But here we can preparing a specific medicine will be used for a specific person. That is the main aim of precision medicine, and that can be done by using AI. Then cancer biomarkers. Cancer biomarkers, this is an indicator that is found in blood or other body fluids which can be detected or diagnosed by predicting that those biomarker, we can identify the type of cancer. There are various kind of biomarkers are there. For example, one is diagnostic biomarker. The diagnostic biomarker is to identify the presence of cancer, like prostate cancer or some other cancer is there or not. Then prognostic biomarkers are there. They indicate how aggressive the disease may be. Then predictive biomarkers are there. In that predictive biomarkers, predict the response to a specific therapy. For example, HER2 in breast cancer for targeted therapy. Then monitoring biomarkers, track treatment effectiveness or recurrence, like colorectal cancer. So these are the opportunities which is produced by the AI, but some limitations or challenges are there. That is bias and generalizability. Bias, when this biasness happen, for example, when the data or the method used are not representative of the whole patient population, as I told. That time, that AI is producing a generalized data, not individual data. For example, the AI model is trained mostly in data from one ethnic group, or it may not perform well for other. That is the limitation. That leads to unfair or inaccurate predictions, or misdiagnosis, or unequal treatment outcomes. Generalizability, what is that? That is the ability of a model or finding to work well across different patient groups, settings, and populations. A model with high this general form can be applied broadly beyond the specific data set was trained on. For example, I'm telling that a cancer biomarker validated across multiple population and clinical settings. So that means a specific biomarker is needed. So that bias limits fairness and accuracy of the data. And this generalized data ensure reliable, wide application research and AI tool in cancer care. Next one is the immune therapy. Immune therapy actually is a highly complex method. For example, dynamic interactions between tumor, immune system, patient unique biology. These are the various variations which cannot be detected by AI, as it involves in very complex genetic things. Here the AI struggles. Immune response very widely across the patient, means the immune response varies one person to another person. One drug which is given to a patient, it may vary, its effect may vary to other patients. So this variability across the patient is one of the major concern. Then the tumor which is produced, their microenvironment, their complexity, they are very much heterogeneous. So identifying those things, these complex things by AI is become difficult. Then multiomics and clinical data are very much become massive. They are collecting lots of data. As they are confused, they are producing noises and difficult to integrate those data to the actual or real data. That is the problem. AI tools and model used in cancer immunotherapy. There are various AI tools like Scorpio, Loris, HistoTME explained. Okay. So these are the things that the main data source is routine. For example, Scorpio, that is routine blood test data. Loris standard blood test data plus tumor multinational bodies and digital pathology slides for history. This Scorpio, it is an AI framework which integrates like multiomics, clinical data to predict outcome in cancer, immunotherapy, and identifying the potential biomarker. It is the main action that is produced Then Loris, another one. This is another platform where they are managing or analyzing the large scale imaging and clinical data sets. They are producing more precise, more accurate imaging. They are from that specific image. Normally, a human being is not able to identify the explanation of specific images. That can be more clarified and result oriented by using. We can produce more result oriented by using this AI technology. HistoTME is a deep learning tool actually to analyze the histopathology slides. Like to study the tumor microenvironments, identifying the immune cell types, which kind of immune cell is activated, which kind of immune cell is depressed, like that. Their spatial pattern and immunotherapy research. So in recent advancement and future direction, I can tell you that it can predict immunotherapy responses by analyzing various kind of data. It can personalize the treatment planning to a specific patient, discovering the novel target area by analyzing the various protein structures and genes, genomics and proteomics. They are reducing the duration which is needed to develop a specific drug. Then optimizing a radiotherapy and overcoming the regulatory hurdles that we have to think about that how this regulatory requirement or regulatory hurdles can be overcome. Data interpretation can be developed or can be improved, and clinical data integration, advancing precision medicine, all these things are needed to produce more and more accurate data and to serve the patient more accurately and more precisely. Now, risk and disadvantages associated with AI. As I told you that AI can work and AI can reduce the effort, human effort. So that leads to decrease in number of jobs that may leads. It will, the human efforts to create machine that can think are huge threat to the existence of the human race. That quotation is given by a very renowned scientist, Stephen Hawking. High cost creation of AI requires huge cost and they are very complex machines. It will produce unemployment also. AI can cause unemployment as things would be automated, reducing the need for human labor. But always think that once the type of job is changing, not it producing the unemployment. So for that, we have to, every time we have to be more updated about this, all the technologies which is used in humankind. For example, we are using Android phones. So that Android phone operating system require a revision. Every time it revised and it will get updated. Then only your mobile is useful. Otherwise not. The same here. We have to learn AI. We have to update it as the day progresses. and we have to know the application of AI, how it can be applied and all this. So one thing is that the AI is developed by the human brain and the intelligence. AI following the datas, they are not intelligent like human beings. The data, what they are getting, data, what are the inputs are given to them. According to that data, they are predicting a specific thing. But it is not, but human intelligence doesn't depend upon this type of data. Is a part of data, but not a whole. We can think differently. We have emotions and we can predict also. And we can learn from experience. We are not machine. We are learning from different types of experience, and according to experience, we are performing, not like AI. And humans also have a creativity, but the AI don't have. So the humans are always better unless and until if we are using this AI only for our support, only for the development of treatment, development of other things. If we are making AI equal to human being or better than human being, then it leads to problem, otherwise not. So if we want to survive for longer time, we have to consider AI and we have to utilize AI in different developmental processes. Finally, we can conclude that it is a very big thing in pharmaceutical industry. In R&D also, it will increase the success rate, drug development process, reduce the duration of drug development process. Also, it gives a strategic advantages. It can transform the immunotherapy of cancer. It can accelerate the process. It can identify the different targets. It can personalize medicine. It can produce a enhanced real-time monitoring. And finally, the challenges such as interpretability, data standardized, and regulatory barriers remains AI's growing in precision medicine, positioning it as a powerful tool to maximize the effectiveness of immunotherapy and improve patient survival and the quality of life. So thank you everyone for listening me. This is my university, Maulana Abul Kalam Azad University of Technology. And these are my few students. Among them, few students helped me to prepare this slide also. Thank you to them also. And thank you all.
Thank you so much, sir. That was a very wonderful session regarding the use of artificial intelligence in pharmaceutical technology. So now I would like to request the participants if they are having any questions or queries regarding the event. So they can ask in the chat box or they can directly ask the speaker, sir. All the participants, if you are having any queries or questions regarding this event, you can ask in the chat box. Okay, sir. Some of the questions which I have received from our YouTube streaming. So the first question is, What challenges exist in getting regulatory approval for AI-based tools in pharma?
What kind of challenges?
What
Regulatory.
challenges exist in getting regulatory approval for AI-based tools in pharma?
The data, what we are getting, actually, the data we are collecting, the collection procedure, how the data are collected. In that case, there is no specific regulatory rules. That is the main limitation.
Okay, sir. So another question is,
For collecting data, using those datas and interpretation of the datas, a specific rules and regulation is needed very much in regulatory.
okay, sir. So another question is, sir, how can AI help in herbal medicine drug discovery?
Herbal medicine discovery. The process is same that the AI which is used actually, that normally the herbal medicine is nothing but the plant active material identification and separation of those. So it takes longer time when humans are using. Normally when the humans are using those, collecting that plant and extracting that plant, and finally we are identifying their effects, either that specific active constituent is it can be targeted for a specific receptor or specific protein. So these things, manually these things takes longer time. But when we are using this AI technology, we can predict all these things. Like nowadays it is using network pharmacology we are using, and so many other AI tools we are using for identifying the protein structure, to identifying the targets. Okay.
Thank you, sir. So another question is, what kind of patient data is essential for AI-based personalized immunotherapy approaches?
What kind of patient data?
Yes, sir.
Normally, what kind of data means? The normal data. The data, for example, the genetic data of that specific person, the location of the person, where they are from, their age, sex, is there any other drugs they are taking or not, or is there any liver toxicity or liver damage, or for example, that is kidney damage. These things are there or not. This information is very much needed. Means AI is running only on data. So the data, whatever the data we are getting, if we are incorporating those datas, then only the final outcome will come.
Thank you very much, sir. Proceeding further, I would like to request Professor Dr. Subhasis Devnath, sir, Principal of Bharat Pharmaceutical Technology, to present the certificate to our speaker, sir, Dr. Sandeepan Das Gupta, sir, virtually.
1 minute, sir.
Yeah. Thank you very much, sir, for your nice presentation. And it was really, I feel, it will be very helpful for the participants and also for our faculty members. The way you have explained the AI in cancer therapy, it was really good. And from behalf of Bharat Pharmaceutical Technology, I really thank you, because today is a festival day, but then also you have taken time. Even in our college also, the puja is going on. So you have taken time and like accepted our invitation. Pralay sir, if you will share the certificate.
Thank you, Principal Sir. Thank you very much. Again, I am telling that thank you for giving me this opportunity. Still, I am also learning what is AI and how it is applied to pharmaceutical fields. So every one of us are under learning stage.
Yes, sir. As it is a new,
Uh,
I think we must learn from each other. So kindly accept the certificate virtually, and
Thank you, sir.
thank you again, sir, from our side and the organizing team. Thank you very much.
Thank you so much, sir. Thank you so much.
Thank you very much, sir. So now I would like to request Mr. Santosh Ganguly, sir, Assistant Professor of Pharmaceutical Technology, to kindly deliver the vote of thanks.
Thank you very much, Bhaskar sir. I hope I'm audible.
Yes, sir, you are audible, sir.
Okay. To begin with, a very good afternoon to all. It is indeed an honor and a privilege to deliver the vote of thanks at the conclusion of today's illuminating lecture. On behalf of the organizing committee, I extend our deepest gratitude to Dr. Sandipan Dasgupta sir for his erudite presentation on artificial intelligence in pharmaceutical technology and revolutionizing cancer immunotherapy. Sir, your scholarly discourse has provided us with profound insights into the transformative role of artificial intelligence in accelerating pharmaceutical innovation and in shaping the future of precision oncology. The clarity with which you articulated complex concepts, coupled with the depth of your expertise, has greatly enriched our understanding of this rapidly evolving field. Your lecture has not only broadened our intellectual horizons, but also underscored the immense potential of interdisciplinary research in addressing some of the most pressing challenges in healthcare system today. We are truly grateful for the time and effort you devoted to sharing your knowledge with us today. I also wish to acknowledge the organizing and supporting teams for their meticulous effort in making this session possible. And I would also like to thank our learned audience for their attentive participation and engagement. Once again, we express our sincere appreciation and gratitude to you, Dr. Sandipan Dasgupta sir, for your valuable contribution to this academic gathering. Thank you.
Thank you, sir.
Thank you so much, sir. Now I cordially thank all the participants for their valuable presence. We will share the evaluation and feedback link in the WhatsApp group, and you are requested to kindly fill up today. So thank you very much for attending the program. Thank you so much.
Thank you very much, sir.
Thank you, sir.
Looking forward to meet you someday physically.
Sure. My pleasure. My pleasure.
All the participants, please note that we have one more session today. So please join by 2 o'clock. And thank you, Sudhirmal sir, also for connecting us.
Thank you, sir. So I'm leaving, sir.
Yeah, yes, sir. Yes, sir.
Oh, okay. Thank you.
Good day, sir.
Good night, sir.
Stop.
audible?
Am I audible?
Yes, sir.
Yes, sir.
Audible.
You're audible.
Is the slide is visible?
Yes, sir. Visible, sir.
Namaskar. A very good afternoon, everyone. So, after a successful completion of first session of day 3 of our EIFTP program, we have stepped into the second session of the program. It gives an immense pleasure to welcome our esteemed guests, eminent speakers, respected faculty members, and active participants to this EIFTP program, which is entitled as Immersive Technologies and Artificial Intelligence in Pharmacy Education. Means building the future of learning. Truly been organized by Bharat Pharmaceutical Technology, Agartala. On behalf of organizing committee of Bharat Pharmaceutical Technology, I, Ms. Mokrisha Singh, Assistant Professor, heartily welcome you all once again. We are extremely fortuitous today to have with us an incredible speaker, Dr. Parthapratim Roy Sir, Associate Professor, Department of Pharmaceutical and Medicinal Chemistry, Institute of Pharmacy, Guru Ghasidas, Central University, Bilaspur. So now I request Mr. Sunil, Associate Professor of Bharat Pharmaceutical Technology, kindly grace our speaker.
May I start?
Mr. Sunirmal Bhattacharya, sir. Please kindly grace our speaker.
Yes.
Thank you, Motoshi ma'am. It's give
May I
me,
start?
sir, give me 1 second. It's give me immense pleasure to, yes, am I audible?
Yes, yes.
Yes, sir.
It's give me immense pleasure to extend a warm and cordial welcome to our esteemed speaker. Sir, give me 1 second. It give me immense pleasure to, yes, am I audible?
Yes.
Immense pleasure to extend a warm and cordial welcome to our esteemed speaker for today's session. He is none other than Dr. Parthapratim Roy. Dr. Parthapratim Roy is presently working as Associate Professor in the Department of Pharmacy, Guru Ghasidas Vishwavidyalaya, Bilaspur. He did his B.Pharm and M.Pharm from Jadavpur University, Kolkata. He obtained his PhD from Jadavpur University, Kolkata, under the guidance of Professor Kunal Roy. He did his postdoctoral study in European project Cadastre from University of Insubria, Italy, under the supervision of Professor Paola Grammatica and Esther Papa. His PhD dissertation topic was in silico modeling of selected classes of cytochrome inhibitors using chemoretic tools. He worked as a postdoctoral fellow in European project registered in University of Insubria, Italy, in the topic of development and validation of QSR models on emerging organic pollutants. His field of interest in silico drug design, ADME, and toxicity modeling. Dr. Partho Pratim Roy is expert in the development and validation of models based on quantitative structure, QSR, to predict toxicological and ecotoxicological endpoints for conventional and emerging pollutants, agrochemicals, etc. Dr. Roy has published several more than 60 research papers in peer-reviewed national and international journals. He presented over 25 scientific contributions to national and international congress. He has successfully completed the research project from UGC and DST served during these years. He is the recipient of the Outstanding Faculty in Health and Medical Sciences 2019 by Venus International Foundation, Chennai. He was among the top 2% world scientists in list according to the Stanford University survey for the year of 2020, 21, 22, 23, and 24. So it's our great pleasure to hear you from you, sir. The stage is yours, sir. Thank you. By Venus International Foundation, Chennai. He was among the top 2% world scientists in list according to the Stanford University survey for the year of 2020, 21, 22, 23.
Sir, namaskar and welcome. You may start the session, sir. Thank you.
Okay. Thank you.
Yes, sir, you can start the session.
Okay. Thank you, Dr. Bhattacharya, for the nice introduction. Thank you,
Sir,
Professor
namaskar
Devnath.
and welcome. You may start the session.
Okay. Today's topic for this FDP, Immersive Technologies and Artificial Intelligence in Pharmacy Education, Building the Future of Learning. And my discussion will be on artificial intelligence in drug design and toxicity modeling. As we know, drug design and development, it is a costly episode and it is a long-term episode. And it will take around 10 to 12 years and it will cost around $2 billion. So it is a complex process and traditionally expensive. So over the year, some evolution has been taken in the drug discovery process. And in the recent past 2 decades, artificial intelligence that is helping in accelerating drug design and improve other properties of the molecule that needs to be done. So indeed, the development process, it is an integration of chemistry, biology, and computational intelligence. So if we get a brief introduction of the evolution of the drug discovery process from the beginning, we can categorize them into 4 different groups. The first one, that is serendipity, or drug that is discovered by luck. It was the oldest one. And you must know that penicillin, cycloserine, this type of drugs, they are come into the market through accident. Then after this, we have some chemical modification for the drug design and development. And one example is stated here, salicylic acid to acetyl salicylic acid. Simple modification. Third one is screening. Screening of the compounds by trial and error method. So it is repetitive. We are trialing several compounds. So up to these 3, serendipity, chemical modification, and screening, there is no rational basis for the drug design. But the demand nowadays, you need to give proper explanation of the biochemical pathway, mechanism of action, and the targets associated with the drug for the rational drug design. If we look the complexity of the drug discovery process. So initially, we don't require any kind of explanation. We tested a chemical and it shows some activity. Yes, it can be approved as a drug candidate. But over the time, the requirements or restriction for the development of drug is increasing. So this is a snapshot where what are the properties we need to modify. So initially, we need a valid biochemical hypothesis. And it should be targeted because drug action that is purely on the selectivity. You need a target. It should be selective. It should be potent and some other pharmacokinetic property, physically stable, soluble, permeable, metabolically stable. It should be non-inducing. It should be reversible, durable, and finally some toxicity parameter that has to be carried out to get a drug before clinical trial. So all these episodes in experimentation that is not possible. So from 10,000 compounds, if we are lucky, we can get one drug molecule. So what to do with this? That is why we need computer-aided drug design. So this is the segment in the drug discovery process and the timeline. So initially, what we used to do from the disease to clinical trial, it will take around 8 years. From target identification, target validation, lead discovery, lead optimization, and up to preclinical trial. So it will take around 8 years. But with the advent of this computer-aided drug design or in the exponential rise in the computational power, we can reduce the step into several months. So we are nothing but simply reducing the time of the drug development process. That is why nowadays drugs are being marketed within few years. So for target identification, we have some bioinformatics tools, homology modeling, reverse docking. Target validation, we have some tools, computational tools. Lead discovery, mainly virtual screening through pharmacophore or docking. Lead optimization, QSAR, molecular dynamics, molecular mechanics, and docking. And some toxicity parameter, ADMET prediction, and off-target effect of the drug that can be analyzed prior to synthesis. So this is nothing, indeed a high throughput screening part in the drug discovery process. Now, what is artificial intelligence? It is artificial, as the name indicates that it is mimicking something. Or intelligence, we are giving some capacity to learn or to solve some problems. So indeed, this can be defined that the capacity given to a machine by human to memorize and learn from the experience to create or to judge or make a decision, etc. Nowadays also we are getting the same because online market is now very famous. And I used to get the notification of the components that I used to search. So the website is trained with my thoughts and he used to supply me what I need or what makes me good. So this is artificial intelligence. Different approaches are being applied in drug discovery process, categorized into 4 groups. Machine learning, this mainly used regression and classification and QSAR modeling. This is a non-supervised model. Deep learning, that is supervised models that uses neural networks, mainly natural language processing. This is mining the biomedical literatures. That is a tool that is being used to study the literatures and to get the idea, to get fundamentals for an objective of the study. And nowadays, student, teachers, all are using ChatGPT is very famous. Also, we are using Quillboard, plagiarism tools, and some Grammarly, and also different publishers. They are also providing Gen AI in Elsevier and other publishing house. So these are some tools that is related to mainly bibliography or publication or textual content. Another is reinforcement learning, that is optimization of the molecular generation, lead generation by AI tools. AI, ML, and deep learning, these are the subtypes. AI, that is a technique that can enable a machine to mimic human behavior. And machine learning, it is a subset of AI. As discussed, it uses statistical tools, mainly regression and classification-based models, to enable machine to improve the experience. It has a limitation because if error occurs, it has to be fixed by human, means it is user-defined. And in some deep learning, it is a subset of machine learning that uses supervised tools, mainly neural networks, to optimize a solution from a problem. And in that case, it rectifies its errors because it is an iterative process. In neural network, if you know, we are minimizing the error all the times. So it repetitive. It is an iterative process. Until the error is minimized, the process gets repeated. So it cannot be, it fix its own error. So this is the advantage. So AI growth, it is a literature data, the first one, which is collected from Scopus, and it indeed indicates that from the last decades, the exponential increase in the AI tools, and mainly in the biochemistry, medicine, and chemistry, these three major parts are being applied, mainly AI tools, mainly to drug discovery. And surprisingly, these graphs are generated from Scopus. They are also using AI to generate this graph. These are some literature that also indicate the revolutionary power of your artificial intelligence in the drug discovery in various highly reputed journals. So that is why there is a paradigm shift from your traditional conventional method of drug discovery to computer-aided or AI-assisted drug discovery. And the parameters here given, time, animal cost, laboratory requirement, animal house, reproducibility, types of chemical. These are the parameters. So in all the parameters, for time, in conventional, it will time consume, and computer aided, it is required less time. Animal sacrifice, no. Cost less. Laboratory units, just a high figure configuration computers. Animal house, no. Reproducibility, yes. Chemicals, nahi chahiye. And 3R principle is strictly followed in the computer aided drug design. So this shift driven us to use artificial intelligence in the integrative pathway of the drug discovery. The role, basically, role of AI in not only in drug discovery, in other fields also, as we are confined in the drug discovery pathway. So mainly we can segment into 5 groups. So in target identification and validation, de novo drug design, AI generated molecules, drug repurposing, it is a well-known phenomena occurring this time, ADMET prediction, and clinical trial optimization of the selected candidate. Now, this is the recent pipeline of the drug discovery as everybody is going to follow. The computer-assisted rational drug design pathway. So initially, we have to select a target. This is very, very important for the prospect of drug discovery, as we need a basic biochemical pathway and we need a target that is associated with this. So to select a target, what are the computational tools that are being used? Reverse docking, protein structure modeling, and bioinformatics. Hopefully, I don't know how many of you know about docking and reverse docking. So simply docking is nothing, fitting of your molecule into the receptor site using the principle of lock and key arrangement and also confirm induced fit model, mainly induced fit model. So what used to do, we used to see the binding pattern or interaction of the molecule with the receptor. And in reverse docking, the opposite phenomena occurs. Mainly, we are using a single ligand and we are using multiple receptor. And we are trying to fit all the ligand into the receptor site. And we are getting the score for ligand in each of every target. And the high ranked receptor or target can be prioritized. Now, the second step is target validation. Now, I have selected a target. And now I have to see whether the target is druggable or not. Means whether it is linked to some biological process or some disease or not. It has to be validated. So we have some computational tools that can be used like आपका string, BLAST, cytoscope. These are the tools that are AI tools mainly that are being used. And one practical aspect that has to be taken into account, that is, we are selecting a target, but we are not able to crystallize the target. Because in SBDD, if you follow that, we are getting the structure of the protein from PDB, protein data bank. So the method is X-ray crystallography or NMR spectroscopy. So we have to select those targets for the protein can be experimentally extracted through crystallization techniques. So after target selection for a specific disease and target validation, next step is lead discovery. Lead discovery means we are finding some lead molecules that can produce the biological action through drug receptor interaction. So for this, because nowadays the digital repositories for the chemical that is increasing. We have PubChem, we have Zinc, we have DrugBank, and so many. So what we can do, we can screen the compound through molecular docking. Because my target is selected. I have selected my receptor. Now I am placing all the compound library in my receptor side, and accordingly I am getting the rank of the compound through binding interaction. So that is one thing. Another thing is structure-based pharmacophore. From the co-crystal structure, we are generating a pharmacophore. Pharmacophore is nothing but a three-dimensional arrangement of the property of the molecules. So, after placing the pharmacophore of the screen compound, we are getting some best match. Compounds we are matching with the pharmacophore. Because initial drug discovery concept was like that similar molecule will produce similar activity. But nowadays, the concept has been changed. This change is due to the pharmacophore modeling, because we need some essential feature, means core structure, that is responsible for biological activity. So it can be present in diverse structure. That is why diverse structure can produce similar activity. That is the concept in pharmacophore modeling. So with docking and pharmacophore, we are getting some lead molecules. Then we have to optimize our lead molecule. Means we get the lead molecules and we are redocking our molecules. And we are getting the binding interaction score as well as QSAR from quantitative structure activity relationship. We are getting the theoretical predicted values of each compounds. So as a rule of thumb, we used to say if the predicted activity value is less than 1 nanomolar, in nanomolar range, we can treat that this molecule can be treated as a drug candidate for the further study. And if not, then we have to repeat the process of optimizing or placing the fragments in the specific scaffold and iterate the process until your activity will go into nanomolar range. Nowadays, one other phenomena that is being widely practiced, that is molecular dynamics solution. That is quite necessary nowadays, which actually give information of the stability of your complex, compactness through a period of time when your complex is heated or in adverse condition. So if all the criteria fulfills, then we can go to preclinical studies. Means we need to see the ADMET prediction and physiologically based pharmacokinetic solution, in which we used to see different absorption, metabolism, and toxicity parameters. And based on these, we can also filter some compound. And after filtration, the best heat will be produced to your clinical trials. So this is a overall pathway in recent times we are following to get a heat compound for clinical trials. Okay. Now, as I already described, this part, target selection and validation, it is most critical steps, and it involves in identifying biological targets, which could be protein, receptor, enzyme, or gene. Target selection ensure molecules is druggable or suitable for the further study. So these are the softwares. All are, you can say, AI, or they are digital repositories. So softwares or tools for target selection, that is protein data bank. Whenever we used to see protein data bank, we search our proteins. For example, aromatase, UIP19, a lots of protein will appear in the sheet. And from there, we used to see Homo sapiens, we used to see resolution, we used to see X-ray diffraction, we used to see Ramachandran plot. So these are some restrictions that are being added for the selection which target to take. BLAST, cluster omega, it is used for sequence similarity and homology modeling in case we don't have any protein. So we need to make a protein. So homology modeling is a widely used technique. Only one software that is available, LUDI, in which 30% of if sequence similarity is there, then we can develop a protein from the sequence. And nowadays, AlphaFold 2, probably you know, in last year, the scientist got Nobel Prize for AlphaFold 2, which for theoretical prediction of protein. And modeler, Swiss modeler, that is a completely AI tools, web-based tools. Now, after selection of these target from these sources, we have to validate. Means we need to see whether these targets has any relevance to the biological action. Cytoscope, that is a open source tool, AI tools. It's mainly depict the biological means signaling or mapping pathway, biological pathway of any protein or any biological actions that is freely available. Another database, string. It mainly involves in protein-protein interaction. Means you have several proteins or you are giving some sequence and you are looking at the other protein sequence. Means this protein has any interaction with other protein, and combinedly it will give which signaling pathway or which biological process that is being occurring through your protein. Another is IPA, Ingenuity Pathway Analysis, that is not freely available. That is also for pathway-based validation, means biological pathway, means biological hypothesis, pathway that is validated through this artificial intelligence tool. And these are the snapshot of this tool. Probably you can go to the website and you will see the game. Okay. So my target is selected through this tool and they are validated. And now I am quite confident that yes, this target is associated with this disease. So lead discovery, as I told earlier, that we practice mainly two aspects, and molecular docking, another is pharmacophore modeling, and third one is QSR based screening. So this is one example that is given. I am not elaborating this because remdesivir, that is an antiviral via virtual screening against RNA polymerase. Here the target is RNA polymerase. And the SAR of or structural features that is present in the molecules are like this: a phosphate groups and hydrophobic tail and a sugar moiety and a base. So these are the features that is necessary. Initial search was carried out using these features. And finally, yes, we are lucky and we got this as a lead compounds or as a heat compounds, and it is now in market. So what we are screening this Jing database, mainly Campbell, PubChem, also Drug Bank, also for natural products, Coconut database, NCBI database. So we have a lot of database, and in the next slide, we will mention the number of sources. So if we categorize the sources in 4 formats. structural database, chemical database, biological database, and chemical property database. You will see amples of data. For structural database, we have PDB, binding DB, drug bank. These are the structural database which give information to your structural property or proteins. Chemical database that is quite common in QSAR analysis. It includes Zinc, NCBI, PubChem, ChemSpider, and ChemID Plus, and many more. Biological database, PubChem also giving a biological database. Nowadays, all the databasing are merged together and they are giving in PubChem also. And property database, ChemSpider, PubChem. Whenever we search for a molecule in PubChem, we just write the name, and you will see in the toolbar, it gives all information of the compound from its IUPAC name, physical property, toxicological property, biological property, and many more. So all are there. Yes, these slides highlight that how many compounds are in different databases. Campbell has nearly more than 2 million compounds. ChemDB nearly 5 million compounds. Coconut, that is for natural products, it is more than 4 lakh. Drug gene interaction database, it has 40,000 genes, 10,000 drugs, and possible of 1 lakh interaction. Drug bank is in fewer class, in around 15,000. PubChem, 119 million compounds. 327 million substrate, 296 million bioactivities. Right? So this is big. PDB, more than 2 lakh of structures. So this is huge. And it is stated that more than 10 to the power 60 chemical, biological, and other repositories are in the databases. So manually, we cannot handle these things. So our hope is only artificial intelligence, because we need to filter. Now, the one point that is kept in the mind for artificial intelligence analysis means accuracy of the input data. That is very, very much important in data mining studies. As we are stressing that our target should be validated, we have to be very much careful that our structure is also validated. You might get a database or data set of around 200 compounds, suppose, for example, original data. So chemical curation that is necessary. Initially, what we have to do, we have to see whether any duplicates that exist in our data. That actually happens. We are extracting data in SMILE format, and different data source will give different SMILE for the same compound. So duplicates should be there. We are getting isomers. They are duplicates. So initially we have to erase these duplicates. Then analysis of intra and inter-lab experimental validity. That is one of the major points that has to be considered, because different lab uses different condition and different reference standard. So we cannot club all that. This is defined in OECD guideline 2. OECD guideline 1, it should be have a defined endpoint. Means mixing is not possible. Exclusion of unreliable data source means whenever we search, because we are getting a data from a database and they are mentioning something. Yes, this data is from this study and they are giving the reference paper. In some data, you will not be given with some reference points. Means there is no reference paper for this data. So we cannot take those data. We have to delete these. Detection and verification of activity clip, some data or some structures that are not validated and they are showing they are odd man out type, means outlier. So we have to remove that also. And calculation of modelability index for mainly this is practiced in QSAR studies. We have to calculate that the rule of thumb is that your data activity range should be within or more than 3 log unit. So if your data activity range is 1 log unit, you are not able to get a model. That is quite sure. Right? And a consensus QSAR prediction say we can delete some compounds and identification and correction of mislabeled compound. That is also possible. So all the step has to be carried out carefully before going to further studies. Because we have validated our targets, now we have to validate our data also. That is why in the database, you are getting nearly 6 lakh compound. So initially what we used to do, we used to study the duplicates. Then we used to add Lipinski filter of 5. So then after we used to add ADMET filter. So these are the curations that are being, so after all that curation, you have to analyze this also. So finally, you will get your curated data and evidence, ample evidence of paper. They are showing that if you curate the data, your prediction reliability will be increased. That is for sure. Some papers of Alexander Tropsha that is reported. Okay. So we got our protein, we got our data, and we get the lead compounds as described earlier. Right? So approaches SAR, QSAR, molecular modeling, molecular dynamics, and ADMET properties. All we have already discussed this. And yes, this is one drug, this is one example, that is imatinib. That is a optimized lead. Optimization is done for kinase inhibition. Kinase is an enzyme. So it is a structure-based optimization of the imatinib structure from the previous one. And another drug, oseltamivir, that is antiviral drug from natural sources, that is also optimized by improved bioavailability. Means ADMET property. We are looking at the ADMET property and we are changing the substituent and we got finally oseltamivir. So there is a lot of other drugs also. I am giving these two examples to you. Okay. Now, this is the framework for ligand-based, the data source or collection and curation. These are the source, Campbell, PubChem, drug bank, compounds and structure. So after curation, we are going for the next part, means QSAR studies. Hopefully, I don't know, all of you might be knowing QSAR. It is in definition, it is a mathematical relationship between your structure and biological activity. And structure that is represented by their properties mainly. Or in our terms, we used to say the property as our descriptors. 0D and 1D, mainly simple molecular formula, number of atom counts. These are 0D, 1D properties. 2D, some connectivity indices. And in 3D, you will find some other shadow indices and solvent, accessible surface area, molar refractivity. These kind of parameters are being calculated for the structures. Because, and we are trying to maintain the representation of the descriptor, that whole molecule can be represented. Because in drug action, we need both hydrophilic and lipophilic portions. So all the parts means log P and polar surface area that is in combination. Then as usual, this is machine learning tools. As I explained, they use regression and classification based model. So after getting the activity and the molecular representation or structure from to get their properties, and now we are correlating the properties with the structure. And in regression, you will get a quantitative value of each structure, and classification, you will get the class where active or inactive. And so after getting the heat or lead compounds, AI application mainly used for toxicity prediction, de novo drug design, and other application. This is mainly the ligand-based drug discovery pathway where AI is being implemented. In structure-based drug discovery, we have mainly target structure prediction, that is from the sequence if your target is not there, or from the sequence for the biological mapping, means biological pathway analysis. Then de novo drug design, this is quite widely used right now. Based on the structure of the receptor, we are generating fragments and we are linking the fragments to get new molecules. All the AI techniques, that is a neural network techniques mainly, black box methods, drug target binding affinity. That is mainly the Ki value we are getting. This is nothing but implementation of molecular docking in AI tools. We are getting the affinity, means binding interaction of the drug, and we are scoring the drugs. And drug target interaction. So we have different targets. We have drug. This is some encoding feature, numerical representation, and we are getting 0 or 1, means whether it is hitting the target or it is not hitting the target. So mainly these tools are being applied in SBDD, structure based, if we are considering the structure for the development of the drug. That is already described. QSAR in drug design, molecular docking, hopefully all you know. This is one figure that we are placing the fragments in the molecules. That is somehow it is de novo practice. It is a tailoring process. And nowadays, the important steps they are asking all the reviewer or paper on all the studies, they are asking for molecular dynamic simulation. Dynamics is something a physical movement of the atom over a time. Means in that case, we are hitting the compound complex in the protein structure and 300 Kelvin, and we are looking at the deflection in the protein structure. So the pathway is like that: obtaining a molecular structure, setting main parameters, giving corresponding molecular force field. The force field are like AMBER, GROMEX, RED-MD or Gaussian, NMDA. These are the force fields that are being applied. This depends and in which GROMEX is freely available, anybody can use, and other is also implemented in commercial softwares. So setting the box size and pH of the values, optimizing the system energy. And finally, you will get some RMSD plot, RMSF plot, RG analysis, and hydrogen bond interaction plot. So this RMSE plot that will give that the stability of the complex, means you can see that is very small. The blue one that is stable, means it is not deflected over the time, 100 nanosecond. RMSA plot that is give the idea of the compactness of the complex, whether it is compact or not. Right? In addition, we have also linear free landscape energy, all are many other parameters that is also being used. Another is important parameter, that is PCA cluster analysis. So in that case, we are analyzing the complex of our heat compound, and the complex of the co-crystal. And we are observing whether the cluster in our case is far away, going outside or inside. This also gives the stability and compactness of the study. So these are some lead optimization tool. Shorinja, this is a commercial GROMACS that is freely available. OECD QSAR toolbox that is freely available. EMBARK that is also not freely available. DTC lab tools for mainly ligand-based drug discovery, QSAR type analysis, and QSAR-INS that is also free on request, DTC. So we have amples of free softwares. We need not to go for the commercial one. So finally, the last step is the preclinical trials. So it was observed in 1990s, late 1990s, that 90% of the drug candidates, they are failed in the clinical trials because of the poor pharmacokinetic properties. 80% of the reason is pharmacokinetic property, poor pharmacokinetic property. That is why we are using the ADMET analysis, the physiologically based pharmacokinetic property analysis prior to experimentation, means prior to synthesis and clinical trial. So this is indeed the theoretical part of the drug discovery process and AI tools, where we can incorporate our AI tools. So now I will discuss some tools that is being used in physiochemical property analysis, toxicity analysis, as well as in drug means targets selection. So in this series, ADMET Lab 3.0, that is AI tools, that is free. Absorption, distribution, metabolism, and toxicity, physical and medicinal chemistry aspects also. If this is one of the snapshot of the output for this compound, so you will get this page. If you want, I can go to the, so you will get the physiochemical properties like molecular weight, log P, TPSA, and medicinal chemistry aspects that is also given, that is SA score, means it will indicate your compound is easily synthesizable, synthesizability index. Absorption, Caco2 cell permeability, distribution, whether it is protein bound, metabolism, which enzymes going to metabolize your compound, whether cytochrome or other enzymes. Excretion, you will get a roughly idea of T-half, ADME, and you will get the toxicity information and some points. And it will also give your toxicity pathway also, in which pathway it will become toxic. Another important aspects in this Pro and ADMET Lab 3, this structure is found toxic for certain endpoints. For example, hepatotoxicity. So AI, artificial intelligence incorporated in ADMET 3 will give the points indicated here, these points, also this point, these points. So these fragments is responsible for this kind of toxicity. So indeed, it was a backward information, means if we get this information, we can modify these fragments also. So this is very, very interesting. So if you want, I can go to its website. Okay. No. Yes, this is it visible? So this is AdmitLab 3. If you want to start ADMET evaluation, you can start over here. It will get started. Simply, you have to put the smile, or another option
Sir,
is draw a molecule.
uh,
Hmm,
sir,
Okay.
I want, sorry for interruption. Actually, the web page is
Yes.
not visible, sir.
Web page is not visible. Okay, I have to.
You have to share Windows complete PC, I think.
Okay. So, yes. Windows. Which one? Anjali, complete Windows कहाँ पे शब्द? I'm sorry.
No problem, sir.
Yeah. Okay, now it is visible.
Yes, sir. Yes, sir,
Okay.
it's visible. Thank you, sir.
So simply you have to draw a molecule or enter a smile. I don't know I am having a smile or not. Uh, Simply I am copying the smile of your tamoxifen in my ADMET lab, and you have to submit. So you will get all these physiochemical properties as described. All these you can get. So whatever the property you need, you can take those. Okay. Okay. Now slide is visible.
No, sir, it's not visible.
Slide is not visible. So okay, I need to do what to do. Okay. Now,
Yes, sir.
now it is visible. Okay. So next tool is SCA, similarity ensemble approach. That is mainly used for the selection of the target. Means here also you have to give the smile notation, and based on your structure, it will give the probability of affinity towards different targets. This is one target selection tool. Secondarily, another target selection. I am not going into online because I have to switch to your presentation as well as online. Because all are available, you can try. So in pass online also, we can select the smile and get the prediction. So in the lower side, you will see that acetylcholine M3 receptor agonist. It is giving some score. So these are the targets. So based on the target from various input site. Target Fisher, another target selection tool. That is mainly designed for your MAO-B inhibitor, monoamino oxidase B inhibitor. So in this case, you have to go for a registration, and then you can also, this is a xanthine type of molecules. And yes, these are the targets, and we are going to see that target monoamine oxidase B and monoamine oxidase A are there. Another tool, AI tools, that is CODD predicted. That is a combo package mainly. It will identify the target, it will give the bioactivation, also it will give the lead optimization. What you have to do, you have to draw or you have to give input of your SMILE notation of the compound. This is also freely available. If you want, I can go to the site also, but it will another also switching to your stop screening to another is your Swiss ADME. That is a old one and most widely used because it has several packages: Swiss doc, Swiss farm, Swiss sidechain predictor, bioisosteric replacement for lead optimization, target prediction, ADMET, and Swiss similarity. All the modules that are available in Swiss ADME. So in that case also, you have to draw the structure or you can enter the SMILE. You will get the output. Swiss target, as I showed earlier for this xanthine molecule, I used the same molecule in Swiss target. Yes, in earlier case, acetylcholine was the first prior target. In Swiss target also, it is the first ranked target. So, in my opinion, in order to validate a target, you can apply more than 2 or 3 AI tools and you can compare the results, whether all these AI tools are giving similar output or not. That is a validity and reproducibility of the AI tools that has to be carried out to get the user confidence enough to go for further studies. This is also a free tool, ProTox 3.0. This is a toxicity tool for small molecules. In ProTox, we have different types of toxicity: liver toxicity, other organ toxicity, some pathway, nucleic acid pathway toxicity, some acute toxicity, all are given. Actually, I need to show you the ProTox. Is it visible?
Yes, sir. Visible.
So this is the website where the compound is tamoxifen. You know, so from the SMILE or from the structure, you can get the structure and you will see that what are the toxicity. Categorize organ toxicity, hepatotoxicity, neurotoxicity, nephro, respiratory, and cardiotoxicity. All are there. Toxicity endpoint, in which we have carcinogenicity, blood-brain barrier, ecotoxicity, clinical toxicity, and nutritional toxicity. Nuclear receptor signaling pathway, is there any toxicity related to this? Stress response pathway, also different toxicity. Molecule inhibiting events, means that is biological events, whether thyroid hormone receptor alpha initiating or different, and metabolism part. Here for the ease of demonstration, I am giving hepatotoxicity and neurotoxicity, and just going to start prediction. Okay. So you will get the output like this. Predicted LD50 is 1190 milligram per kg, and predicted toxicity class is 4. So here is the color 1, 2, 3. They are toxic, means highly toxic, moderately toxic, least toxic, and they are non-toxic. If 6, the compound is non-toxic. So it is in class 4. So we can say that our molecule is not toxic. Another important thing, that average similarity, because in artificial intelligence, some in-house model is there based on some chemicals, large database, and read across nowadays a widely used tools that search for structural similarity. So average similarity also is 100%, means I have similar type of compounds in my training set. So it will give also the prediction accuracy 100%. So we are confident enough with reliability of the results. It is also giving some receptor that is modulating your tamoxifen, mainly androgen receptor, average similarity known ligands 91.46%. So it is an androgen receptor antagonist, you know. So this is a simple tools giving very good results. Okay, I am not going to the slide. I will go to other tools if I am having. Yes, COT. As I mentioned earlier, COD, it is a combo package. For example, I am going to the target prediction, for which I am giving, it is a pre-loaded smile that is for donepezil. I know donepezil is a antagonist of acetylcholinesterase. So after giving the smile notation, I simply go for start prediction. So this is your molecule, and you will see acetylcholinesterase, and relevant score is maximum 0.71, and the next is kinase 0.0062. So it is a highly selective acetylcholinesterase inhibitor. So, and you can get the bioactivity prediction. The input method for this also, we can use smile of the same compound, donepezil. Choose a target. If you are looking for a compound, your target is not known, you can try. These are the targets that is available. So ACE, my target. I use the best method, and I would like to get the prediction for this. So you will get a predicted value of this compound. So these are very simple tools, but giving very insightful results. I don't know, I am having other tools or not. No, that is okay. So going back to the slide. Okay, the slide is visible?
Yes, sir.
Yes,
The
sir.
slide is Okay, okay, thank you. So these are the tools, AI tools I described, that is for ADMET, target selection and some toxicity parameters. We have also tools for library generation, that is for lead optimization. Right? So the first one is MOLOP, now a widely used AI tools for bioisosteric replacement. Right? That is also Zandrag, that is also free. Anybody can use this. So for example, this is my input file and I am highlighting the part where the bioisosteric replacement has to be occurred. So from this to you will get another compound, 5 members isoxazole for this furan compound. So in that case also you have to provide the smile. And another thing that you can manually select the replacement point or allow the software to choose the favorable point because it has AI tools for selecting the points for bioisosteric replacement. So both options are available. In Zandrag also, a structure-based fragment replacement for the generation of the new lead molecule can be done. So in doing so, initially you have to choose a protein and ligand file. You have to select a protein and ligand file you have to upload. That is very easy, and in server you can do this. Then this is your ligand and these are the points where the fragment replacement will going to occur. So the server presents all replaceable substructure on the input ligand. Please select the substructure you want to modify and click the submit button for 2 minutes. So it is very fast. So you are giving the protein, you are giving the ligand. So after inputting these two files, the server will show you that this is your ligands and we find the possible points where you can do your replacement based on the drug receptor study. So you know that this part is essential and this part can be replaced by these fragments. So manually you can click on the options they are giving. And finally, you will get the new generated lead compound from the fragments. This is very easy. Another lead generation tool is Smiley. That is also available in software version, also in AI version. To do this, initially you need to identify the core structure or the pharmacophore, means which core structure needs to be present. Okay? And what are the substituted? Means to be changed. This can be done by both QSAR and molecular docking studies. From the molecular docking study, you will get the interaction pattern and which part of the molecule interacting with which amino acids. And from the QSAR study, you can identify the pattern or properties of the molecule that is important, and this property confine part in the molecular structure. So this is a corroborative, means combined class feature. So after finalizing that, this is my core structure. For example, beta blocker, aryl amino propanol, aryl oxypropanol amine. This is the core structure. And what are the modification? The modification is terminal nitrogen, tertiary amine, or in the first phenyl, it can be naphthyl, or in selective beta blockers, there is para substitution in the aromatic ring. So we know the SAR part. So after doing docking, pharmacophore, and Q studies, you have the idea. This is my core structure, and these are the replacement that favor or disfavor the study. So you need to select the core structure. You need to select the substituent. And another thing, you need to add a linking molecule, linker. How to attach or your replacement will be attached directly to the core structure, or you need some linker, aliphatic side chain or some other thing. So after doing all these things, you can run your sample. Here the input is a smile, or they will give the desired format, and this is also very easy. From this you can generate number of molecules for the optimization of the lead. This is commercial reinvent, this using generative reinforcement to generate new molecules, neural network. This is your input molecule, and you just input the molecule, you will get a score. If it is okay, then you have to go for further step. If it is not okay, the process will iterate. These are commercial, not available. CREM is chemically reasonable mutations. It will find chemical replacement framework. As CODD prediction does, this is also same. You are giving your scaffold and it will look for the suitable substituent in the as a fragments in the molecules. Mainly de novo drug design, it is being used. Chemistry 42, another drug generation. This is also commercial. Mole drug AI system. This is also commercial. So mainly your AI in a nutshell, if we see the applications in ligand-based drug discovery, in data curation, physicochemical properties, de novo drug design, virtual static toxicity prediction, bioisosteres generation, etc. In SBDD, structure-based drug design, initially to protein structure prediction, protein drug interaction, target selection, de novo drug design, polypharmacology. It is now a well-known study for multifactorial disease. AI has tremendous application over there. We have different success stories of AI, HIV protease inhibitor, Indinavir, haloperidol, and some carbonic anhydrase inhibitor, renin inhibitor, dehydrofolase reductase inhibitor, neuramidase inhibitor, and HIV transcriptase. So all are structure-based application of AI to get a successful candidate. Also, we have many more thymidine synthase, collagenase, phospholipase A2, glycogen phosphorylase. So I have already completed my lecture, but we need to be careful some points to use these AI tools, and some challenges also, some limitations are also AI. The first step, as I mentioned earlier, availability of high quality data. That is the necessary facts, because in chemical, we have to curate the data. Also in structure, we are getting a protein with missing some residue. That is not accurate structure. We have to get high quality of input data. As mentioned in a famous paper, if you are going to give a garbage in, you will get a garbage out. So you have to give precise high quality data to get high quality and reliable input. Another is it is a black box method, AI, artificial intelligence. You don't know what is happening inside. So another problem is lack of interpretability. How to interpret? So this is a crucial task that has to be handled by the user. Another is availability and accessibility of the models. Because reproducibility, you are giving a results whether I used your AI tool or you use the tool, the output will be same. So for commercial software, availability and accessibility is not possible. But for the free softwares, that is good. Another is multi-target drug design, that is polypharmacology approach, in which target selectivity is a very problem that causes, means whether you have selected 3 targets. So inter-target affinity prediction means acetylcholine, for example, acetylcholine, butyrylcholine, esterase, and monoamine oxidase. I have selected these 3 targets for Alzheimer's disease for a compound. So inter-target affinity prediction for this compound for these 3 targets has to be done, because we have to maintain the order, whether this target is highly selective for this or highly selected for this or equal selectivity. As you know, selectivity is must, but less selectivity will not impart the biological action. So these are the points that has to be kept in the mind whenever you are applying the artificial intelligence in drug discovery. These are the softwares that is available, as I already described many of them for the drug discovery and toxicity prediction. Okay, this is my lab, Laboratory of Drug Discovery and Ecotoxicology. And these are some publication. Mainly, we are dealing with ecotoxicity modeling, QSAR based modeling, and we are doing some Alzheimer research and anti-cancer drug therapy. So for toxicity modeling, basically we are using the QSAR based approach, and for the designing we are using SBDD ligand based approach, and also we are also doing some synthetic part. So I need to acknowledge my university, Guru Ghasidas Vishwavidyalaya, my whole team. It is a big team, and all the works that is done with the help of this team. Also, I want to acknowledge Bharat Pharmaceutical Technology, Agartala, Tripura, for inviting me to give a lecture on artificial intelligence in toxicity and drug discovery. So, if you have any queries or any queries, you can ask me, or if you need some further help, you can contact me. Thank you.
Thank you, sir, for such informative session. Now, to all the participants, if you are having any queries, you can drop your questions in the chat box, or you can directly ask to our speaker, sir. So there is some questions from participants.
Okay, SCA tool.
Please explain CA rule tool.
So I can also, once again. SCA means single approach. So once again, I need to share. SCA, now it is visible. SCA, similarity ensemble search. It is mainly used for the search of the target. As in other cases, I have given the smile notation. And for example, this smile. Copy. So I am simply copying the smile and try SCA. Similarity ensemble search. So based on your input, okay. So this is our Donepezil. So based on the input, it identifies the targets. And it will give the score. 1 means highly affinity towards this target. Right? So this is nothing but like a Swiss target prediction, target Fisher, or CODD PRED, like this a artificial intelligence tool to select the target for a supplied ligand. Is it okay?
Okay, sir. Thank you.
Thank you, sir. Sir, you have a next question from Mr. Pralay Malik. If we are using any AI tool, do we need to validate the method?
Yes.
Okay, sir. Thank you.
Because validation is important. As I said that AI tools that are giving similar output. Similar output means we are getting our targets for a specified compound. So as I said, you need to do this for 2 or 3 AI tools, and you need to see whether the output is same or similar in trend. So then we can see that yes, the algorithm that is being used in different tools giving a similar output, that has to be validated, because we have many options. So we need to check whether the output is same or not.
Thank you, sir. We have another question from Mr. Aminul Islam. Can we get the list of web tools named for future reference?
List of?
Can we get the list of web tools named for future reference?
Yes, I have given all which I am working for all their things for docking, switch dock. Autodock is also free. For target selection, these are all AI tools. Means target Fisher, SEA, switch target prediction, and many more. For ADMET, you can AdmetLab 3. Also, you can Protox, also giving ADMET properties. This kind of artificial intelligence tools are being used, regularly used.
Thank you, sir. Another last question from Dr. Ashmita Saha, ma'am. Can we use the predictive toxicity software for drug discovery, subsiding animal toxicity studies?
Yes,
Can
that
we
is
use the predictive toxicity software?
that is done. That is now it is a widely practiced. That is being adopted by your REACH and other regulations in the West. They are using the theoretical activity for the compound that are toxic. That has to be toxic, and REACH that requires theoretical toxicity prediction whenever a new compound is going to be registered. Although experimental validation is the ultimate one, because whenever you find a chemical that is highly toxic in theoretical studies, so final validation is your experimentation. But rather doing experimentation on thousands of moiety, you can do experimentation of a single moiety. So this is the power of computational power, because we are reducing, but we cannot completely exclude experimentation. That has to be done, right? For some exclusion of some compounds, for restriction of some compounds, that is done.
Thank you, sir. Now, any participants having any queries? Thank you, sir. So now moving towards next, I would like to request Professor Dr. Subhasis Devnath, sir, Principal of Bharat Pharmaceutical Technology, to present the certificate to our speaker, sir, Dr. Partha Pratim Roy, virtually.
Namaskar, sir.
Good afternoon, sir.
A wonderful presentation, and I hope not only you have given a presentation, but also you have but beneficial for the participants and also our faculties. It was really an excellent presentation, sir, and I request you to accept the certificate virtually from our side. Please accept, sir.
I am accepting. Thank you very much,
Thank
Professor
you, sir.
Devnath.
Yes, sir. And I hope we'll meet physically also one day, so that we can interact more.
Yes,
Yes.
definitely. And from the participant, anybody require any help, they can mail me. So I am always there to help.
Thank you. Thank you very much, sir.
Thank you.
Thank you, sir.
Thank you, Mr. Dr. Professor Mollik, Ashmita Madam, and others also. I do not remember the name, for the process and for inviting me and giving me the chance. Thank you very much. And I wish the success of this EFDP.
Thank you, sir.
Thank
Now
you.
I
Thank you.
request Dr. Shantanu Malik, sir, Associate Professor of Bharat Pharmaceutical Technology, to deliver vote of thanks.
Thank you, madam. Am I audible?
Yes, sir, you are audible.
Yeah, okay. Good afternoon to everyone present here today, our honorable guest speaker, respected principal, sir, and esteemed faculty members from various institutions across the country. On behalf of the organizing committee, I sincerely thank Dr. Partha Pratim Roy, sir, for your insightful session on artificial intelligence in drug design and toxicity. In today's world, where rapid and precise drug discovery is vital, your talk highlighted how artificial intelligence can revolutionize the pharmaceutical research, reduce the development time, and enhance safety by predicting toxicity with greater accuracy. This perspective is truly relevant as we strive to address global health challenges efficiently. I hope the tools you suggested today will definitely help the faculty members to carry out their research projects. I also extend my gratitude to our faculty colleagues for their active engagement, to the organizing team for their dedication, and our institution for fostering such enriching academic exchanges. Thank you once again, Dr. Roy, sir, for you sharing your expertise and guiding us towards future-ready pharmaceutical education. Thank you very much, sir.
Thank you. Thank
madam.
you.
Thank you, sir. Now I cordially thanks all the participants for your valuable presence. We will share the evaluation feedback link in the WhatsApp group, and you are requested to submit it by today. Thank you. Have a wonderful day.